Hello everyone. I'm really fortunate to sit here with Professor Dan Burke, Emeritus Professor of Pharmaceutical Metabolism and expert in cancer research. Welcome to South Africa.
Thank you. Thank you.
Please tell me more about your work in cancer and your research that you've done that led you here today lecturing on integrative treatment and options on cancer management.
Goes back a long way. My first experiments on cancer research was as a postdoctoral scientist in Dallas back in 1973.
Sure.
And I started, the boss of the lab put me as one of his team onto looking at why do certain chemicals cause cancer. And that's all linked to the same mechanisms that detoxify pharmaceutical drugs. And
so you're talking about smoking chemicals,
yeah,
and petrochemicals?
Yeah, very much so. The sort of chemicals you get in the diesel particle fumes and the smoke. Right. And yeah, it's the main reason why cigarettes cause lung cancer.
So that's that exact area. Why would that chemical then switch on a cancer gene?
That's for the molecular biologists. We were more concerned, entirely concerned, with these we call them PAHs. And they're actually, in the initial state, pretty harmless. The body turns them into something noxious. It makes little chemical changes to their structure, very specific changes. Mm-hmm. And it's the derivative made by the body itself that causes the cancer. And that's what we were looking at. And we were trying to identify which particular enzymes, because it's all enzymatic processes. Sure. There are enzymes in the body, and they cause this to happen, and how do they do it, and what are the changes in the structure. And that really was the hot area in cancer research in those days. Nowadays, it's because of new technology and new knowledge, it's more about the molecular biology, the aberrant genes that cause cancer. So we were more concerned in why they become carcinogenic.
Right. Right. And what did you discover?
What we discovered was the role of a particular family of enzymes called the cytochromes P450, or nowadays we'd call them the CYP enzyme.
The CYP1B1.
Yeah, it's
a protein enzyme, is that
yeah,
right?
all enzymes are proteins. Enzymes are functional proteins. In those days, I mean, it's very interesting. The whole family of enzymes. Now they know there are more than 8,000 of them across all forms of life, from the smallest to us. But in those days, there was right at the start. You know, you're very fortunate if you get in at the start of a field. Mm-hmm. They were only discovered in 1964. A couple of Japanese scientists, and they gave them the name. Right. And when I started my PhD on them in England, there was thought to be only one. And it was a conundrum, because enzymes are very specific. The old idea about enzymes was one enzyme, one substrate, one particular chemical that it will work on and no other. And then it rapidly became realized that many pharmaceutical drugs, pharmaceutical medicine compounds are worked on by this enzyme, and so there was only two possible consequences. Either there were lots of them, which we now know to be the case, and each of them is quite specific, or it was what we would call an incredibly bendy enzyme. It could modify its configuration, and that's where I started. And during the time of my sort of formative years as a scientist, gradually they discovered more and more of these enzymes, and now we know there are thousands of them.
That's right. So you're talking about the molecular structure of the CYP1B1 enzyme being flexible enough to fit into different molecules.
That would have been one of the original CYP enzymes that were known about, and that was the concept. Was it able to handle lots of different types of structured pharmaceuticals, because its active site. Active site's a bit like a docking, you know, the docking site, say for rockets on
Yes.
on the space station. That was one concept that at the time was the best, that it would change its shape to accommodate.
Right.
They do, now it's realized it's not so much that, it's more the fact there's a lot of different ones of these CYP enzymes. There's about 35 of them in humans, and there's about, say, 8,000 more than out there. So it's often the case, you know, at the beginning of a scientific area you think one thing and you work it to death, and then you find out actually it was really something else.
It's all part of the process. I suppose you have to start somewhere.
Yeah, I think it was Haldane, who said, JBS Haldane, physicist, once said that whenever he had completed a research project, he realized he should have been looking at a different project. You do this. Knowledge is incremental.
The work that you were doing, did that have anything to do with the development of chemotherapy drugs and to try and make them more effective?
In a word, not really as such. The work that we were doing relating to what kind of chemical structural modification happened in these compounds like benzoate to turn them into nasties, into chemicals that cause cancer. That was really more about establishing the kind of structural changes that these CYP cytochrome P450 enzymes could do. And that knowledge was then used later on by lots of people, us, we looked at one specific aspect, to help further understand how the CYP enzymes transform carcinogens. Now your question, funnily enough, was about chemotherapy agents, and they are a class that's still the most clinically used type of anti-cancer drugs. They happen to be a class that is not metabolized by these enzymes I was passionate about.
Right.
Everything, I think, is known about the mechanism of action of chemotherapy, and it has no connection to anything I've ever looked at, be it cancer or anything else.
That's interesting.
Yeah, the problem with the chemotherapy drugs is they don't have the right sort of specificity. We could go into that, but it gets complicated.
I'd love to.
You really want to know?
Yes, because if you're looking at the specificity of the chemotherapy drug, I mean, you know, basic knowledge we have is that they target your healthy cells and the cancer
and it's
cells.
why.
Yes, so I think this is where the CYP1B1 research comes in.
It does ultimately. To start with,
give me all the juicy stuff.
Well, I suppose so. I mean, you are talking actually of chalk and cheese. Remember what I said, science develops. And science tends not, great scientists, historians of science have said this, not me. Science doesn't develop like that. It develops like this, and what makes the change is when a really important scientist, or they might not be important at that time, twigs to something and then persuades everyone else that no, it's not like that, it's like that.
That was what happened with you, though, because you discovered the holy grail of cancer research.
We're not that, we were never that important in cancer. There were then and there are now much more important people. But so the chemotherapy drugs, paradoxically, because pretty much has been known about them for a long, long time. The reason why they go for cancer, the reason why they're so imperfect and have all these side effects, what happens to the body, really of all chemicals involved in cancer, they are the least involving the things that I'm interested in, which is these CYP enzymes and what happens. So everything really is known about chemotherapy drugs. I even met, as a young postdoc scientist, I met a couple of the by now old scientists who had invented one of the major classes of chemotherapy. Chemotherapy works in a well-known way, doesn't involve CYP1B1 or any other CYP really, and the flaw in chemotherapy is what it targets. Basically, chemotherapy targets cells that replicate frequently.
And they don't know how to stop. Isn't that
well,
what cancer is in a nutshell?
That would define very succinctly a cancer cell. But the problem with chemotherapy is there's some very important normal cells that in the course of what they do for the body have to replicate frequently. Chemotherapy drugs, because of the way they're designed, they can't distinguish the two modes of frequent replication. In essence, every drug, chemotherapy and anything else, and also natural pharmacological agents, they all have to have a molecular target. And where drugs are nice and specific is where the target is known and the target is confined to the disease state. This is what the pharmaceutical industry uses, is its design paradigm. It looks for targets which were a part of a disease state, but which are confined to the disease state, don't turn up in the healthy. And then it can design a drug. And the chemotherapy, it turns out what it's targeted at are the processes that cause cells to grow and replicate. And the problem is that those targets are not confined to cancer cells. They also turn up in these very important normal cells. The chemotherapy drugs are designed primarily to act instantly and would at their target wherever they find it in the body. So whatever they do to the cancer cell, which is good, unfortunately they do to the normal cell. And that's why you get the side effects. There's a saying which is attributed to that old gunslinger Wyatt Earp, US Marshal in the cowboy days, the hero of gunfight at the OK Corral. Fast is fine, but accuracy is everything. And you can see where he's coming from. If you think about it, it's the essence of pharmacological therapy. If your drug doesn't have the accuracy, that's when you get the side effects. And because of the limitations and what was known in the days of designing and developing the chemotherapy agents, the side effects were inevitable. They weren't meant to happen. The scientists who came up with, designed the chemotherapy drugs, they really thought, and you can understand this, what they were doing was going to be helpful. The problems with chemotherapy, why by and large it is not successful, not in the long term. There are successes. Some types of cancers people can benefit hugely from them, but the problems, they came to light later, and as to why the problems occur, that came to light a bit later. Of course, now we're in the era of targeted cancer drugs. Have you heard this expression?
Yes, I have.
Well, that name alone gives the game away about chemotherapy was untargeted. And whereas the problem with chemotherapy was what turned out to be their targets were not confined to the cancer cells. Now, you know about the Human Genome Project and what it spawned. So this, thanks to the Human Genome Project and what came later, the scientists know now, at last, really for the first time, what's really going on that's going wrong in cancer cells. Why do they act in this terroristic, selfish way? Why do they keep on growing and disrupting the body despite the body saying, no, don't do that? And because the scientists now are getting a grip on understanding at the molecular level which genes have mutated, which are aberrant, what are the proteins then that they're forming, because it's the proteins that matter. You know, the genes make us who we are, the proteins make us tick. What are these aberrant proteins? Why are they aberrant? What's going wrong? Right, now we know this is a protein which is aberrant, it's malfunctioning in the cancer cell, but it's not malfunctioning in the normal cell. Now we can design a drug, and sometimes these are antibodies, sort of proteins to fight proteins. We can design a drug that specifically targets the protein that is wrong in the cancer cell, and wrong in a way we know and understand. For example, often these important chemicals, these proteins in the cancer cell, they regulate the whole process of cell life, death, growth, and they act like traffic lights. There's all this information flying around, and it has to be regulated. And these, they're called signal pathways, signaling proteins, and these signaling proteins are like traffic lights, and they go red and green according to... and in the normal cell, they go red and green as they should, and they respond to each other. Often what happens in the cancer cell, they're either permanently on green, or they're permanently off red. And that's the heart of the problem, they now know. And so they've designed drugs, these targeted drugs, to block them, take them out of process. But there's a problem. These drugs are spectacularly effective for a few months. And then the patient develops resistance.
All right.
And resistance is going to be, shall I stick my neck out?
Go for it.
The last remaining problem for getting the anti-cancer drugs we need.
Are you quite positive? Are you hopeful?
I'm positive that it's the last remaining problem for two reasons. Then we can talk about the hope. The reasons I think it's the last remaining problem, in that I think the problem of which targets, that's now been solved because the cancer scientists now know what they should be looking for. They're very clever people.
I'm sure.
But why resistance is a problem? The science of it predicts, if you read and understand the science, but there is this, one of the most important living cancer researchers, a chap called Professor Burke in the States. He's medically qualified, but he's really a molecular scientist. He leads one of the most important molecular level research groups in the US. Is John Hopkins, I think, in the US. He's also a bit of a heretic, which is why I like him. And in a learned paper he wrote quite recently, he set this out in his opinion, and he's a guru, if you like, that resistance is a problem. And he went further in that paper. So, and it's a year or two ago, so things do change.
Very recent, though.
Very recent in the history of science, 2013 to be precise. And he said, resistance, in his opinion, resistance will be the problem to be solved. And he went so far as to opinion that maybe it's insoluble. That our best efforts at anti-cancer drugs, no matter how targeted, will be defeated by resistance because, well, the cancer cells are clever devils. And the processes that are now understood that cause them to be cancer cells. What is it about a cancer cell? Our normal cells, they grow and they live, and when necessarily they die. For the health, yeah, well, if they need to be replaced, they die, but they don't turn over that often, are healthy cells. But what they do in this regard is to help the health of the body, and they obey the commands of the body. Well, the cancer cells are selfish. They grow only for themselves. They're stupid, of course, because eventually the host dies. And
for sure.
So we don't, but that's,
like a parasite
that's
almost.
Philosophy. You know, we don't get into that. Now, we as a species, have you ever thought, how have we managed to survive as a species all this time, all these, whatever, millions of years? And I think it's gradually coming to light. One of the reasons is called biochemical genetic redundancy. Where we have crucial processes, we don't just have one, and this has been discussed quite recently in science. We have lots of them, so that if one gets accidentally taken out of the process, we've got another one as a backup, and we have a backup and a backup. And that's great for our survival as a species. It's also great for the survival of the cancer cell, it turns out. There are, as you know, thousands of genes in the cell. Well, people like Vogelstein have worked out that it's relatively few, probably less than 200 of these genes that are really crucially involved in controlling, driving the growth and death of cells that are crucial in cancer. And the cancer cell turns out to have redundancy in these crucial processes. So when a clever new targeted anti-cancer drug takes one out, the cancer cell, clever little devil that it is, just turns on another process that's always there. It might have been doing something else. It switches it. We don't fully know. And that's the point at which
the potential is always sitting
the
there.
cancer cell has now become resistant. So the oncologist switches to a new of the targeted anti-cancer drug. They've all got different ones for different targets. And after a while, the cancer cell develops resistance to that by kicking in yet another of the processes. And Vogelstein was of the opinion in 2013 in his paper that this may actually defeat the best of which we can. Now, I know that you are very interested in integrative therapies.
Yes, I am. And I think that you just listening to you, just reinforces the fact that we need to take our health back and do something about it as much as we can ourselves with complementary approaches, be it, improving sleep, improving quality of life, reducing stress, optimizing nutrition, drinking lots of water, reducing toxicity around you, and then hopefully, putting products into the body that are extracted also scientifically to optimize the deficiency in nutrients from our food.
Yeah, and I have the benefit, I'm very lucky, very privileged that I've seen this from now both sides of the coin. 30 years as a member of the scientific pharmacology establishment, if you like, very small part of the cancer research establishment, albeit in universities rather than the industry, but entirely seeing everything from that angle, over the years becoming more and more impressed by what was known of the pharmacology of natural compounds, and then being able, after retirement, to switch. I've if you like, a gamekeeper turned poacher. Some of my old scientific friends say I've gone to the dark side.
Oh my
and
goodness.
now
I hope they're still friends of yours.
Some of them no longer will communicate with me.
Goodness.
And now I'm involved entirely from health, from the perspective of the natural plant-made chemicals, what we call phytochemicals.
I just want to go back on that. So we're talking the difference between synthetically produced compounds versus natural product extracted compounds.
Yes, entirely. I say that we scientists are clever, but Professor Nature is actually an even cleverer scientist. But what I'd like to do is just finish my train, if I may, of what I was coming to, because it links the natural products to the cancer molecular science. This problem of resistance. And let me use maybe a metaphor. You're driving down the motorway, nice and fast, on your own, and then suddenly you see up ahead all the traffic has come to a halt. There's some blockage, but you see it before you get there. You've got a good sat-nav or a good navigator with a map. What will you probably do at that point?
Slam on brakes.
Okay, and then what will you do?
Wait in traffic.
You see this coming a lot. You'll take an alternative route.
If there's an art, for sure.
If you can, you'll take an alternative route. You will bypass the block. Think about it. That's exactly what the clever cancer cell is doing when it's developing resistance to the brand new targeted anti-cancer drugs by switching in an alternative mechanism. And this is where the natural compounds come in. And I know you're interested in curcumin. It's not my main topic, but
yes,
I have developed a very great respect for curcumin. And I've looked into now for several years its pharmacology. There's a very good book, by the way, for anyone who's not a scientist, who wants to understand cancer and especially chemotherapy. There's a book published about 5 years ago called Cancer, the Emperor of Maladies. I think you can see why it's called that. It's by a doctor in America. The more I learned about the pharmacology of curcumin, the anti-cancer mechanisms of curcumin, the more and more I came to regard it. The higher I came to regard it, and I might even use that terminology and call curcumin, the emperor of anti-cancer phytochemicals.
Oh my goodness.
Yeah.
Would you put it above cannabinoids? Would you
Oh,
put
yes.
it above Salvestrols?
Oh yes, in one particular respect. I don't think it's productive, really, personally, to enter in these comparisons, because they're each of what you've mentioned, cannabinoids, the active chemical, Salvestrols, of course, the active chemicals in green tea, in garlic, in broccoli, all of these, they're all very powerful pharmacologically in their own right. So it's not that one is superior to the other. But the state of knowledge of each of them is that curcumin alone, it has been identified as having so many different important molecular targets in cancer cells that it's like an arsenal all of its own. If I remind you that the likely problem that we're going to face once we have all the new targeted anti-cancer drugs is the resistance that develops. Of all the natural phytochemicals, anti-cancer phytochemicals that have been identified and researched, curcumin alone has the mass of different targets. It can block all those alternative bypass roads that you can take. There are at least 100 different molecular targets to do with cancer that curcumin just... And the amazing thing is all the identified cancer targets of curcumin so far seem only to be therapeutic for us. And maybe that's happened by trial and error. I used to have to teach toxicology to the medical students. I enjoyed teaching it. And I'd ask them, what's the longest clinical trial in human existence? The answer is probably our diet. Sure. We eat what's good for us and we tend to avoid what's going to kill us and be harmful, and that's trial and error. And I think maybe that's why curcumin, we've ended up sticking with curcumin in the form of turmeric. You eat it as turmeric in your cooking. Because it turns out to be good in this chronic inflammatory process, which we now know is underlying so many different diseases, and not to cause too many problems.
I would love to just understand more about exactly the role and the mechanism of action of the biocurcumin extract in the treatment of this, or not the treatment of it, but in opening the blockages that develops during the resistance process of a chemotherapy drug.
Yeah. So if you take these bypasses, if you take the alternative bypass roads when you see the block happening on the motorway, so what I've been trying to explain to medical doctors treating cancer oncologists is maybe if you give curcumin alongside your clever new targeted anti-cancer drugs, just maybe the curcumin, by blocking many of the alternative resistance pathways in the cancer cell, might lessen the chance, slow down the development of resistance. There are two...
That's where the hope is.
That's what I'm thinking. Now, I've not been spectacularly effective convincing oncologists of this, especially in the UK, where they really don't like alternative complementary therapies. But there are, I know, 2 doctors in Munich who I've talked to who reached this conclusion on their own. So there are doctors out there. So they
would look at doing trials?
They will, and it won't just be curcumin, but curcumin is the best of them right now.
Right. Tell me a little bit about Salvestrols. I know we're running out of time. You're going up Table Mountain. You're only here for a short time.
And it's a lovely day.
Sunny South Africa. Beautiful day. I won't keep you, but I just want to know, because from your introduction to me was this is the man to interview about Salvestrols. Okay.
Well, our cancer research carried on, of course, as a theme, and my interest in these CYP enzymes carried on. And in the early 1990s, I was trying to map out CYP enzymes in various human tissues. I was working with the pathologists at the University of Aberdeen, and why doing this? Because I thought, well, maybe it will help us understand tissue-specific side effects of drugs.
Right. So you're talking apoptosis, you're talking about the chemical, the chemotherapy killing off and the die-off of
well,
the cells?
I was at that. I wasn't even looking at cancer to start with. The reason so many drugs cause side effects is that often it is their derivative made by CYP enzymes.
Right.
It is their
Right.
metabolite that's toxic.
Okay.
For example, paracetamol.
Okay.
So I thought, well, if we know more about how they map out into different tissues, we'll understand why sometimes the side effects are tissue specific. And so I teamed up with the pathologists and they taught me a great deal. And we started using their techniques, and we were able to start mapping CYP enzymes. And then, because of the methodology we developed, we made a chance discovery. I think it was Szent Georgiou, a very famous Hungarian biochemist from my days as an undergraduate. We were taught about it. I think he said something along the lines, discovery is seeing what everyone else has seen but no one else has recognized.
Lovely.
Yeah, in science the things come across like
Sure.
that, and if you have a fleeting chance of recognizing them, if you don't, that discovery is gone.
Yes.
And we realized there was something wrong with our results. We were seeing something that could only be described as a type of CYP enzyme of a certain subfamily, a certain clan, if you like, we're talking Scots, that seemed to be present only in cancer cells. We couldn't detect it in the normal cells.
So they were not in normal tissue
It didn't
cells?
appear to be.
Right.
We now know that there is a little of it, but not much.
Okay.
And yet its characteristics meant it couldn't be any of the then known CYP enzymes. And we knew, because of our methodology, of certain antibody methodology, it was of a certain clan, subfamily, certain group. And we even wrote grants to the main granting bodies for cancer and for
This is now you and Professor Jerry Potter.
Well, he came along later with Jerry. Yeah,
Right.
Jerry's the second important stage. So on this one, with my as he is now, Professor Graham Murray, he started off as my PhD student. He was eventually Professor of Pathology at Aberdeen. We wrote grant applications to the main cancer and medical research funding agency saying, well, we think there might be, this might be this. And I wish I'd kept copies of the letter. Of course they turned us down, we were heretics. And I remember one of the letters from the referee said, it's well known there's only 2 members of this particular subfamily of CYP enzymes, and the applicants are claiming that there must be a third. Well, now we know it is the third. Yes. Fortunately, at that time, Professor Bill Greenlee in the States, he and his team discovered the existence of CYP1B1 using new type of DNA-related methodology. We were protein people. I'm a protein person. That's old-fashioned now. And so just at the right time, because luck comes into it in discovery, Exactly at the right time, he announced his discovery. He then graciously consented to collaborate with us, provided us with the tools that he had that we needed, that we could nail this to this CYP1B1. Then I left Aberdeen at that point. I was appointed head of Leicester School of Pharmacy down in England, and took with me this idea that what we now knew about CYP1B1, it's an enzyme of a type that specializes in changing the chemical structures of drug-like molecules. It's the precise chemical structure of a drug that determines which targets it hits, what effects it has. If you change that structure, you can change its effects.
Right.
And you only need a small change. It has to be very specific. In theory then, CYP1B1 should be able to change an inactive compound to an active anti-cancer compound.
Right.
And in the case of CYP1B1, because it's not quite exclusive to cancer cells, we now know, but there's a lot of it in cancer cells, little or none in normal cells. That would happen almost exclusively in cancer cells. No side effects, because whatever happened in the cancer cell stayed in the cancer cell. Right. And that type of compound is called a prodrug. You design something to be inactive, but which can then be, you design it so it can be activated by a specific process in a specific place in the proximity of its target. Right. CYP1B1 would do that. Okay. So we set out to do that. And my next bit of luck, I was able to attract Professor, wasn't professor then, Jerry Potter, to the team. And he already had a successful track of developing, well, he designed actually, some anti-cancer drugs that are very important clinically. I mean, there's this new, relatively still new, anti-prostate cancer drug. It's branded as Zytiga, but pharmacologically it's called abiraterone. That was his, long before we joined forces. Anyway, I needed a new pharmaceutical chemist on the staff, and he came along, we appointed him, explained the idea for the prodrug. He'd had some similar ideas of his own. He took it on board and we combined forces, and he designed and made these compounds, because that's the skills of a medicinal chemist.
Correct. So you complemented each other beautifully.
Yeah, me and my team, we tested them, and indeed we did come up with a prototype, new type of anti-cancer pharmaceutical prodrug that was activated by CYP1B1, and we showed this inside the cancer cells. And Jerry went from being doctor to professor in record time. But more importantly, that led us to the Salvestrols, because then we theorized maybe equivalent anti-cancer prodrug molecules might occur naturally in some of our food plants. So we went looking, and in 2002 we found the first of them, and then subsequently we called them Salvestrols. They are indeed nature's natural anti-cancer prodrugs, at least in laboratory.
But these, I followed your research around the discovery of these compounds in food, and it was discovered only in organic food, is that correct?
It's much higher in organic food. We spent, after we'd found them and we decided that we might find a way of helping people benefit from them by extracting the individual Salvestrols and concentrating them in a supplement type of product. We spent the best part of a year also chemically analysing modern sophisticated pharmaceutical style and analytical procedures, chemically analysing all the different fruits and vegetables and herbs we could get our hands on. And indeed we found that Salvestrols as a type are widespread. There are clearly some fruits, vegetables that are much better sources than others. But the big shock was that they weren't present in anything like the abundance we had expected from what we knew of other phytonutrients. Phytonutrient, natural plant-made chemical that supports our health. And so we had, I mean, the Victorian diet, the old Victorian era diet, more and more nutritionists regard as a healthy diet. The advantage also is that science has a pretty good idea of which plants and in what amount were in that diet. We've analysed modern plants for the spectrum and quantity of Salvestrols. We were able to then extrapolate to estimate what range of Salvestrols in what quantity were in the Victorian. And we estimated that our modern diet, at least our modern Western European diet, has less than a fifth, less than 20% probably, of the Salvestrol spectrum and content of the Victorian. And then the question arises why? Because that, we think, puts us at risk.
What did we do to our food?
And what we did to our food, this is where the organic comes in. The main reason for this, clearly, is, well, to understand the answer, you have to know why the plant makes the Salvestrols in the first place. Plants have a very sophisticated chemical defense system against pathogens, against insects, against microbes, but in this case, against fungi. Fungal infection, blight, mold. And it turns out that the Salvestrol compounds are a very important part of a plant's chemical defense against fungal infection. And a key characteristic of the Salvestrols turns out to be that they are made by the plant primarily in direct response to the fungal attack.
So it's a defense mechanism.
So there's a little there normally, fungus hits the plant, biochemistry happens, genes are turned on in the plant, more Salvestrols are made. That puts them squarely into a type of natural chemical called phytoalexins, which are made primarily in response to the problem. you have to be careful because there are lots and lots of phytoalexins. Very, very few of them are Salvestrols. All the Salvestrols we know of are phytoalexins. The vast majority of phytoalexins are not Salvestrols. All dogs are animals, but not all animals are dogs. So if you see something labelled phytoalexin in a herbal store, it's probably not a Salvestrol, not unless we say it is. So the problem then becomes clear. When you spray a food crop with these nasty modern synthetic agrochemical fungicides,
pesticides and fungicides,
specifically fungicides, yeah. Why do you do it? You do it to kill off all the fungi. And if you kill off all the fungi, you remove the very stimulus for the plant to make the Salvestrols.
So they don't need to make it anymore, so they don't
make it
anymore.
Yeah.
But organic plant does not get sprayed, so they keep manufacturing Salvestrols.
And we found they can have up to 30 times higher levels of Salvestrols. And that also goes for many other important phytonutrients as well.
This has been incredibly interesting.
Yeah. There are some other reasons. It's to do with manufacturing of foods and how we cook them, and the fact
that retaining those compounds once you've picked them, obviously storage, you know, whether you freeze them, whether you cook them
how we
to death,
cook
how
them,
you consume
how industry debitters its fruit juices, how
taking out the compounds, I would assume.
Developing sweeter apples and other fruits, and also the simple fact empty calories, well-known concept now. Everyone's stuffing themselves with trashy hamburgers, too many calories, not enough phytonutrition. So it all conspires. I'm often asked, can we get enough Salvestrols for our health purposes from our diet alone? And we think Salvestrols have 3 roles. Prevention, and I wish there was time to talk about the role of prevention in health, but maybe another time.
I'd love to.
Prevention, rescue, and maintenance, which is the name we give to following rescue. You want to keep it that way. We think that if you really worked at it, you could probably get enough Salvestrols from your diet for protection. You'd have to have organically grown, you'd have to eat lots of them, you'd have to make sure they were picked at the right time, etc., etc., etc. But for rescue and for maintenance, for all sorts of reasons, not least the fact that Salvestrols turn up primarily in the outside part of the plant. No problem eating apples.
Which is peeled.
But the oranges, the avocados, not many people eat the peel. So for all sorts of reasons, and including pharmacological dose.
And then you're talking about orthomolecular medicine, actually, when you're looking at the high concentration of supplements that you use to activate a cure, a treatment, not just a maintenance.
We don't use the term cure. You remember, Salvestrols, they have a proven scientific anti-cancer action, mechanism of action we've established beyond all doubt, but they're not a licensed medicine. They must never be described as a cure for cancer. They are part of phytonutrition, helping optimize our diet.
I'd like to finish off by saying that I really wish that we can get this message out to so many people, just simply for them to have another perspective on drug therapy and on the promise and potential hope that we have in using natural compounds together with chemotherapy when it's needed.
Yeah, integrative medicine, part of integrative medicine is the focus on prevention. When we talk about prevention in a cancer-related context, the science is there. I could explain to you, given the time, why you need weekly, almost daily, to do something to help you protect yourself, and you need to start that at an early age. It's no good coming along at the age of 50 and occasionally taking a supplement. The science is there to make it very, very clear that, especially in cancer, you need to be working away at protecting yourself, and even then, you won't necessarily escape the scourge.
It's really sad looking at statistics and the rise in cancer.
Oh,
it's really sad, and especially with children. But we have to end off. You have to get on top of that mountain. And
I do have to
the
get.
Rest of the beautiful
I'm
day.
looking forward to this.
It was such a privilege, and I'm
so
grateful.
Bring me back another time, so that I want to come back.
Absolutely, I'd love that. So
Thank you.
nice to have met you.
Thank you.
Really appreciate your time.
Thank you.
Thanks so much.
Thanks.